so I'm doing a project on therapeutic cloning. Okay, so basically therapeutic cloning is where you create cells for medical research. The aim of it is not to create babies, it's not to produce a clone of an organism. Instead, it's to create lots and lots of stem cells. So a use for it is to be able to repair damaged organs. And the stem cells are basically non-differentiated cells, and then they have an unlimited reproductive potential because those cells, they're not limited by the 40 cell limit as do normal cells. They can infinitely divide until they run out. Okay, so therapeutic cloning and organ repair. So some of our bodily functions even now are using stem cells. So an example of that would be the bone marrow. The bone marrow is generating blood and all the components of blood, like white blood cells and red blood cells that we need. So in case we have an injury, the blood would be differentiated from the bone marrow cells, which are stem cells, and they become the blood cells. However, these cells are really limited because they can only become blood cell types. They can't become cells like nerve cells or brain cells. They can't do that. So the next thing I'm talking about is that some organs are unrepairable and how they can be achieved with therapeutic cloning. So these are some of the organs that are not repairable. So, for example, neural pathways. So because our neural pathways have already entered that G0 state, so that means that they cannot create more nerve cells if they become damaged. So if you suffer a spinal injury, that's why you'll be paralyzed for life, and there's no cure at the moment for it. Well, with therapeutic cloning, it is possible because therapeutic cloning will be able to create stem cells that will be able to differentiate into the nerve cells that you have, and then they'll basically repair the pathways. So an experiment they did was they found that by engineering mice that have broken spinal cords, so basically they can't move their legs, they were able to allow the mouse to regain some motor control by differentiating stem cells into the nerve cells and planting it into the places where the nerves are broken. So some other things are hearts. Obviously, hearts can't reproduce themselves, so if you suffer a heart injury, the only way you can do it right now is to get a transplant, and that's very hard to come by. So again, brain cells. Another thing is insulin cells. So these cells, so basically right now, if you have diabetes, the only way you can continue living is if you receive insulin as an injection, like every day, so it can control your blood sugar levels. And this is because the insulin production facilities in your body are broken, and how they're producing insulin is that they isolate the insulin gene from the human, and they plant it into a bacteria, and then the bacteria produce insulin, and that's how they harvest the insulin right now. But if therapeutic cloning was possible, they'd be able to differentiate healthy insulin-producing cells and plant it into your body and would not cause any problems. So I go on to the third thing is that they can use hybrid cells, hybrid embryos for research. And what these are is that they're basically animal cells, animal embryos from an animal's womb that has the DNA stripped out. And what they do is they inject the human DNA into it, and by doing this, they're able to stop the expression of the animal genes and allow the cell to express human genes. And because these cells have human DNA, they can be guaranteed that the research performed on these will be similar to humans. Although it's not exact, but they can use this sort of cells to conduct research where human cells are not possible. And they function very similar to it. Okay, and the second thing is that they're able to create new organs to replace the malfunctioning organs if it can't repair the damaged organ. So basically in organ transplantation, there are three types of rejections. So these are the three. They're hyperacute, acute, and chronic rejection. So what hyperacute rejection is that the body's immune system immediately recognizes the organ that they're trying to transplant into your body, and then the body basically releases all sorts of antibodies to try and kill it, because they think it's an invader, even though it's not, but they don't know that. This type happened a lot before they were able to identify the patients, so they didn't know that this was going to happen, so when they tried to implant it, the organ often died as the patient is getting transplanted, basically, with the organ. So basically, as they're putting the organ into the patient, they end up with a lump of dead stuff in their hand. So the next thing is acute rejection. This type of rejection happens slower because therefore acute, not hyperacute. So what this happens is that you can be transplanted by the organ perfectly fine. So they'll be able to open your body, place the organ, and they see that there's no problem. So they stitch you up and you can go home. But after a few days, the organ will begin to shut down as your body's immune system recognizes it again. So the last type is chronic rejection. Even if hyperacute rejection and acute rejection don't happen, this will happen anyways. And this is called chronic because obviously it takes a long time for this type of rejection to actually happen. It usually takes many months, but eventually the blood vessels around the organ will die and the blood supply will cut off, and then the organ basically dies after that. So basically all the transplants today will not last you forever, and you will eventually need a new one. So basically, so organ cloning will eliminate this problem because if you were able to, because rejection is primarily caused by the differences in DNA, and because when you have different DNA, the cells on your body have different markers on their surfaces, and that's how the immune system recognizes each cell and determines that it's not a virus or an enemy. So if you were to have the same DNA, then all the organs that were generated from those stem cells would therefore have the same markers, and then your body would recognize it as one of its own instead of as an invader. So this would also eliminate the problem with organ donors, because right now it's very hard for organ donors. For example, a kidney, you'd be giving up a part of your body to save someone else, but then you'd be also missing half of your body's functions. So it's really hard for those people to do that, especially if it's to like give to your brother or something. And then you'd be missing half of it too. But with organ cloning, if you were to be able to create a perfect organ, which is ideal for them, that's what part of the reasons why they even use identical brothers or brothers and sisters, because they would have similar DNA, and with similar DNA, you would not get that rejection problem. So it would eliminate the need for the organ donors if you were able to create them. Okay, so as I mentioned, there's going to be some problems for therapeutic cloning. So one of the first problem is that a large number of eggs required. This type of research is actually very difficult to do. And there's a really low chance of success. So they need a large amount of egg cells to perform research on, like as in human cells, because it's really hard to obtain, and they still need a lot of it. So the solutions for this would be you could either use hybrid embryos, which is really easy to obtain because you can just get it off of animals, and then replace the human DNA. And these types of cells will behave very similar to human cells, and these can be used before on actual human testing when the success rate has actually come out. And the second thing is you can perform the test on animals, because animals share the same kind of function as humans do, because they all use DNA. They all behave similarly, so the test can be performed on animals as they did with those rats, which they tried to fix their nervous systems. So by doing this first, and then moving on to hybrid embryos, they can solve that problem for the lack of eggs. Because right now, in the US, it's legal to get eggs by offering people women money just to get their eggs. It's illegal, so it's really hard for researchers to actually obtain that. And the second thing is that people argue that it's not ethical to clone humans. But in reality, this is not actually technically cloning a human. The identity is not destroyed because the cells that you're actually that you're actually cloning are actually being put back into your own body. So basically there's actually no life that is lost because it's still a part of you. And again, the cells are just extra parts of the body. Say if you have a car and you have parts that are broken, is it wrong to create another part based on that and put it back into the car? So in conclusion, I would just like to say that cloning research must continue, and these medical research benefits are actually very good. And many lives can be saved, especially those organ donors, those people who are waiting in line for organs that are not still getting any after so many months. And the cells are actually a part of you, and these organs can be actually fixed instead of just having to be thrown out, instead of just a minor heart problem. You don't have to replace the organ, you can actually fix it with the stem cells. So, any questions?
Thank you, Xiaolin.